Hi, I'm Lena, Principal Data Scientist at myNEO. myNEO is a company that is focused on developing personalized immunotherapy, and today I want to talk about how we apply natural language processing to cancer genomics. First, I will give an introduction into what are neoantigens and what is immunotherapy, and then I will give an example how we use NLP at myNEO. So what are neoantigens? Neoantigens appear in the context of cancer. Your body is made of cells, and every cell contains DNA, which is the genetic information necessary for the development and function of an organism. It is basically a building template, a biological instruction manual for proteins, which are molecules that play many critical roles in the body. Mutations or mistakes in the DNA sequence can arise, for example, during the replication of cells when they multiply, or when a cell gets damaged, for example, through UV radiation. A cancer cell arises when accumulated mutations are left unrepaired, and that can cause uncontrolled growth and cell proliferation. So basically, mutation or a mistake in the DNA sequence can lead to a mutated and altered protein. In a cell at any given moment, there are proteins produced and proteins broken down and recycled. During this breakdown and recycling process, the big proteins are broken down into small peptides, so small bits of the protein, which can hold the mutated sequence. And the small bits of the protein, the small peptides that do contain the mutated sequence, these we call neoantigens. So then the neoantigens can be presented on the cancer cell's surface, and the immune system can recognize these neoantigens and attack the tumor cell. For that, we have to know that every healthy cell presents a range of peptides all the time from the breakdown process, and only if a cell presents neoantigens, altered peptides that should not be there in a healthy cell, then the immune system reacts to the cell and kills the cell. So what is immunotherapy, and why do we need immunotherapy? So basically, normally the cells function very well and cancer doesn't occur. They catch the abnormal cells before anything happens. But in some cases, the system fails and cancer rises. And in these cases, we then need medical intervention. So immunotherapy is a type of therapy that stimulates the immune system to recognize these molecules, the neoepitopes, and attack the tumor cells. There is a range of different types of immunotherapy, which generally use the power of the body's own immune system to prevent, control, and eliminate cancer. Today, I want to focus on cancer vaccines and immune checkpoint inhibitors. Immune checkpoint inhibitors are a new class of cancer treatments, and they act on a cell pathway which usually prevents the immune system from overreacting to a stimulus. But cancer cells can hijack that regulation mechanism and shut the immune system down. And then a treatment with immune checkpoint inhibitors reverses this silencing of the immune system and reactivates the immune cells. So they can, again, are able to kill the cancer cells. And this generic immunotherapy has shown unprecedented results and led to a breakthrough in cancer therapy. However, it may not work for everyone. There are some patients for which immune checkpoint inhibitors will not provide any benefit, and it's often patients with a low number of mutations. This makes sense because if a patient has many mutations, it will have a lot of neoepitopes that are presented on the cell surface. If the patient does not have to start with many mutations, there will be very few neoantigens actually presented at the cell surface, making it harder for the immune system to detect these cells. So for these patients which do not respond to the non-personalized immunotherapy, they require immunotherapy that specifically boosts the immune system against the tumor cells of this patient. So what happens in this personalized approach, and specifically here I'm talking about cancer vaccines. So first, the patient, the tumor will be screened for targets that are specific to this patient's tumor. So tumor-derived neoantigens. And this happens through many different computational tools, which I will come to in a second. And then once the targets are identified, they can be formulated in a personalized cancer vaccine, which then can prime the immune system of the patient to tackle the cancer by basically painting a big bullseye on the cancer cells by saying, hey, we know how they look, and please attack these cells. So the identification of neoantigens is very crucial, but challenging, as only a subset of mutations and tumor alterations lead to altered proteins. Then these are broken down, and only, again, a subset of the neoepitopes is then presented on the cell surface. So it's quite challenging to identify the neoepitopes that actually make it to the cell surface. And therapy incorporating irrelevant targets can overload the patient's immune system and not be beneficial to therapy. So yeah, it makes sense that in a personalized vaccine, we only want to put in targets that will be effective and not confuse or put in irrelevant targets that only lower the efficacy of the therapy. So how do we then apply NLP to cancer genomics or to a problem in cancer genomics? So first, myNEO, we provide a computational platform for neoantigen discovery and selection. And our computational platform is mainly made of three main parts. So first, we identify possible tumor-specific alterations which lead to neoantigens. So we have a set of potential target neoantigens. Secondly, we identify the neoantigens that make it on the cell surface that are presented. And then in a third part, we screen them for immunogenicity, meaning we check which of these presented neoepitopes are likely to be recognized by the immune system and lead to an immune response. And then we can prioritize our targets and select the best ones for therapy. So today I want to focus on the second part, the neoantigen presentation prediction. Peptides need to be presented at the tumor cell surface to be able to cause an immune response. And the HLAs, which are the genes that encode the MHC molecules, which present the peptides on the cell surface. You will probably throughout the presentation hear me interchangeably use HLA and MHC, which refer either to the gene or the molecule that presents the peptide, but it refers to the same thing. So basically these MHC molecules that present the peptides are very polymorphic. And that means that there are hundreds of different HLA alleles that exist throughout the population, and every individual person has a different set of HLA alleles. This makes the presentation prediction very challenging, as every individual HLA or MHC has a different ligandome, meaning it presents a different set of peptides. Recent increases in the amount of MHC ligandomics data has opened the door to the development of a new generation of presentation prediction algorithms. So what is MS ligandomics? So basically, MS, or mass spectrometry-based ligand discovery, physically detects the peptides which are naturally presented on the cell surface. This is a measure which is more accurate than binding affinity. Binding affinity has been used as a proxy for presentation so far, and it represents the strength of binding between an MHC molecule and a peptide. To measure binding affinity, MHC and the peptide are brought together, and then it is measured how strongly they bind to each other. MS, or mass spectrometry, on the other hand, detects ligands which have been naturally processed to be presented on the MHC. So which have been broken down, have been loaded onto MHC, have made it to the cell surface. So this more accurately represents the biological pathway. And indeed, it has been shown that as little as 5% of tumor-derived peptides predicted by the models to bind the MHC are actually presented when detected by mass spectrometry. So for every patient that you analyze, you then have a set of MHC molecules that are able to present peptides, and you have a set of peptides that you extracted, which you know are presented on these MHC molecules. Both are represented as a sequence of letters equivalent to their sequence of amino acids. So these peptides, proteins, are made out of a chain of amino acids, and this chain can be represented as letters, so one letter for every amino acid. So that means we basically have the biological problem that certain MHC molecules present a group of peptides, and which is different for every HLA in every person. So this problem we can then translate to a natural language processing problem of emphasis on connection within text. To put it as a very simple example, we could think of an MHC as a movie and presented peptides as quotes from that movie, and non-presented peptides as quotes from a different movie or not a movie at all. So either a peptide is related to an MHC, or is related to another MHC, or is maybe not presented by any. So that's a bit how this is translated. So using this data, we developed myNEO-MS, a transformer-like sequence-to-sequence neural network. Data was collected in more than 320,000 peptides eluted from the MHC ligandome of cancer samples and human cell lines, and representing the ligandome of more than 160 different MHC molecules. Peptides and MHCs are ingested as a concatenated string, basically like a sentence, and tokenized at the character level using individual amino acids as tokens. Other tokenizations have also been tried, but they did not improve performance. So basically, because both sentences, so the sentence representing the MHC molecule and the sentence representing the peptide are short enough, so character level tokenization does work well. The model then extracts the important motifs of both peptide and MHC. The model output is the presentation likelihood of a given peptide by a clinically relevant set of MHCs, usually the set of MHCs of a patient. The myNEO-MS model follows a bird-like architecture meaning a bidirectional transformer architecture. It leverages attention mechanisms to focus on the most important input motifs and how they interact. The inputs are always a set of peptides, which are presented or not, and a set of MHC sequences on which they are potentially presented. Positive training data consists of MS-identified peptides, the ones I mentioned, and negative data was synthetically generated. Negative training data outnumbered the positive training data by a 1:50 ratio, as we know that only a very small percentage of all possible peptides that arise from a protein are actually presented on the cell surface. So most peptides will not get presented, and therefore there's quite a ratio between positive and negative data. So on a common test data set, myNEO-MS outperforms currently existing alternatives. Some are ligandomics based, and especially it outperforms the affinity-based methods. And it achieves a precision of 61% at a recall of 40%. myNEO-MS is capable of extrapolation and is able to output precise prediction even for HLA alleles never seen during training. This will minimize the need for additional training data to cover all existing HLA alleles. And this is especially important for rarer HLA alleles where sufficient training data is hard to come by. As not many people express certain HLA alleles, then it's very hard to get enough training data for that specific HLA. We were also able to show that myNEO-MS results are biologically relevant. myNEO-MS prediction correlates well with identification confidence in mass spectrometry experiments. Peptides identified with higher confidence as parts of the MHC ligandome are considered more likely to be presented by myNEO-MS. So that correlates well with each other. And also, myNEO-MS predictions resemble known HLA binding motifs, which we can see here on the lowest graph for one specific HLA allele. So that the peptides that are predicted to be presented, they look very similar to the peptides that are known to be presented by that HLA allele. So in conclusion, we can say that myNEO-MS currently provides accurate and biologically relevant predictions and is a much better alternative for integration into a neoantigen discovery pipeline than affinity-based methods who suffer from high false positive rates. Through more accurate presentation predictions, myNEO-MS can improve neoantigen-driven therapy efficacy. By being able to better select targets, we can decrease the number of ineffective epitopes used in therapy, which is very important to get a strong vaccine. That can also greatly reduce therapy development costs and minimize therapeutic development timelines by limiting the number of validation experiments needed. And also sometimes with cancer patients, you work in very tough timelines to prevent the cancer from developing further. So time is of the essence, and then it is often not possible to perform validation experiments in the lab. So therefore, myNEO-MS for us is an integral part of our pipeline. But we also know, when identifying the best neoantigen targets for therapy, presentation prediction is not the only challenge. So I come back to our prediction pipeline where we have our 3 different modules. So to recap a bit, we not only focus on presentation prediction, but we start with extensive tumor alteration screening. So when we screen the tumor material, we focus on many different types of tumor alterations to identify as many potential targets as possible, which is very important, especially in the patients that have a low number of tumor alterations and which are hard to treat. So we try to find as much as possible. So also for these patients, we can make a good selection. And once we have a confident set of neoantigen targets, we use NeoMS to know which are presented on the cell surface. And then we can further investigate these targets, whether they are likely to cause an immune response. For this purpose, we developed another machine learning tool, NeoIM, which is trained on peptides that have been experimentally validated to be immunogenic, meaning they have been shown before that they cause an immune response. We use the NeoIM model to determine if a neoantigen is likely being recognized by the immune system. And finally, then, for the selection we get, we prioritize them and select the best targets for therapy, which maximize the clinical benefit for the patients. But why stop there? So our next challenge will be to deconvolute the impact that the tumor microenvironment can have on immunotherapy success. And by tumor microenvironment, I mean the presence of different types of immune cells in and around the tumor tissue, which can have stimulating or regulatory effects, and they can have a profound effect on the therapy outcome. So right now we are developing a tumor microenvironment-based prognosis prediction model that links the makeup of the tumor microenvironment to a therapy prognosis. And taken together, these three modeling approaches, with these approaches, we aim to increase the likelihood of therapeutical success. So finally, I hope I was able to give an interesting example of how natural language processing has moved from linguistics into other fields like genetics. And thank you so much for your interest.